All right, everybody, let's talk about case studies 14 and 15, where we're exploring temperature and pH. So again, this is kind of part two of a series where we are examining the growth conditions of these microbes. Last time we looked at oxygen requirements, and this time we're thinking about, wow, how do they handle changes in temperature and pH? Or in reality, which temperature ranges or temperatures and which pHs, should be an R in there, which pHs are allowing these organisms to grow. So what is going on here? Like, why would temperature and pH matter? Well, do they matter to you? Think about that. Can humans live in extreme temperatures? Can humans deal with extreme changes in pH? And just a reminder, pH, which means like potential hydrogen, has to do with the hydrogen ion concentration. In other words, we don't really need to get too technical and things like that, but it has to do with the reactivity of things and molecular stability of some certain proteins and things like that are dependent on pH. In reality, though, what we think of pH is we're thinking of things that are acidic or things that are basic. And things that are acidic, so if we put 7 as like neutral, things that are acidic have a pH less than 7, and things that are basic have a pH greater than 7. Right? And we associate things. So think for a minute, what are some things that are acidic that we think of? Maybe what are some things that are basic? Well, it's things like lemon juice, really strong acids obviously go down here in acidic. Basic, super basic things like oven cleaner, bleach, very, very basic. Okay, but we have this range of 7 in neutral, and then either direction you go, you get more acidic or more basic. And maybe how acidic you are, or how basic the environment is, can dictate whether or not you can live, you can grow or not, if we're speaking about microorganisms. For humans, well, can we think of any parts of our body that might be more or less acidic or basic, right? Our blood is typically a little bit acidic. It's not quite 7. I think it is just a little bit this way. Could be wrong about that, but we're close to the middle, if not a little bit acidic. But what about our stomach, right? Our stomach acid, actually, the acidity of our stomach is right around 2 with all that hydrochloric acid. So inside of our body, not technically inside of our body, but as a part of our system, we have this very acidic environment in our stomach. And then as soon as we get to the intestines, actually, bile salts and all the other things spike that pH back up to around 6. So there's some dynamic pH zones within the space of your body. So it's just something to think about. Inside our cells, however, it's not quite that dynamic. Keep that in mind. All right. So the question again, what are we thinking about here? These are growth conditions. What pHs, what temperatures are microorganisms able to survive and able to grow in? And it does matter. It certainly does matter. So how did we measure this? Well, we took our organisms and we put them in little tubes to grow. Tubes with BHI. And we wanted to see. We didn't use our controls for this. Or I'm sorry, we didn't use our unknowns. We just used some known organisms, some controls. And so we put these different BHI tubes in a variety of temperatures and pHs. So let's say we put Staphylococcus epidermidis inside these, right? S. epidermidis. Staphylococcus epidermidis, common skin bacteria. Let's say we put it in each one of these. Right? We can use a little pipette or we can use a loop, whatever we want. And all of these are BHI. But are we going to grow them at the same temperature? Well, no. Let's say we grew this one at 10 degrees Celsius. Maybe we grew this one at room temperature. Maybe this one at 37 degrees Celsius. Maybe this one at 55 degrees Celsius. So that way, this group of tubes, we can see how well Staph epidermidis, for example, grows at different temperatures. But now we want to do the other side of the lab. Let's talk about pHs. So now we can just do, like, let's say 3, 5, 7, and 9. So we have 2 that are acidic, 1 that is neutral, and 1 that is a little slightly basic. And again, we have that Staph epidermidis in there. But do we want to grow these? What temperature should we grow these at? So that's actually very important, right? Because we don't know which temperature grows best. But again, we know what Staph epidermidis is. It grows on our skin. So we're going to go ahead and do all of these. Oops, I ran out of pen room. All of these, I'm scribbling all over the place. All of these will grow at 37 degrees. Okay, so we put what we would have then done, all of the pH ones go in 37 degrees, and then we have these temperature ones right there. So that is basically the layout of this slab. And again, what is the goal? What is the question we're trying to answer? Well, what temperatures or pHs, what are the growth conditions that allow these microorganisms to grow in? And always think about that question when you're going through these. Well, how do we measure it then? Well, what do you think? How would we measure this? We put these in these tubes. Are we going to do a viable plate count maybe? Maybe, right? That was the milk lab. So are we going to take the tubes and pour them onto a plate, but maybe do a dilution? Zero dilution, right? Because we need to make sure we have it. And then we plate these possible concentrations and then we count them. We could possibly do that because the goal is to see how well they grow at these different temperatures and these different pHs. And the number would certainly tell us that. So if we did that, right, let's talk about the temperature ones. Let's say at 10 degrees, let's say at room temperature, let's say at 37 degrees, let's say at 55. Let's say we did a viable plate count, and at a specific plate concentrate, let's say 10 to the 1, we found 0. Well, that would mean it didn't grow very well. But maybe at room temperature, we had like 10 to the 4. Well, remember, these are negative dilution center. Maybe we had 155. Ooh, that seems like it grew better. But then what if we had 10 to the negative 8? We had 208. So that's not only is this number higher, but this number is way higher. 10 to the 4th higher. And then let's say we have 10 to the 1, negative 1, whatever, just 10 to the 1. We have 0 also. These are just example numbers. So at this point, we count that. We say, hey, the best growth is at that one. We can there determine the growth. But that would be a way to do this. We could certainly do a viable plate count. We're not going to do that. We're not going to do that. Wouldn't be wrong. Wouldn't be a bad idea, but we're not going to do that. Instead, we're going to look at turbidity. So no viable plate count. We're going to look at turbidity. And what is turbidity? Well, that measures the loss of transparency of a liquid. In other words, it going from clear to cloudy. And the turbidity is the loss. Okay? And so what we're going to measure, actively measure, is the absorbance of light. And that absorbance of light is going to tell us the turbidity. And how are we going to do it? Well, we're basically going to shine a flashlight through these tubes. That is really basically what we're doing. But we are doing it with a fancy flashlight called a spectrophotometer. Or a spectrophotometer, however you want to pronounce it. But it's basically a flashlight with some mirrors and a sensor letting you know how much of that light is being absorbed. You can also measure how much is being transmitted, but nope. We're checking out the absorbance of light, basically the turbidity, and we're going to do it using you don't need to know this, but it is a spectrophotometer. Spectrophotometer. And that's what we're doing, putting these in these fancy machines and checking it out. And that's it. And again, always thinking about what was the question. Well, we want to know, how do temperature and pH affect microbial growth? How did we test this? Well, we are going to subject an assortment of microbes to different pHs and temperatures and figure out how much they grow. And what are some ways we could have measured this? Well, we could have done a viable plate count because we are talking about how well it grew, in other words, the number of colonies. But we are going to evaluate using the turbidity of broth. And a tool is spectrophotometer, which would measure the absorbance of light. And that is it. And I will post up some data. And that is it for temperature and pH. Thank you very much.